So there are 3 cortical zones of the cerebellum, and they are the vermal portion, which is just along the midline, the paravermal portion, that's just right next to the midline, and then these lateral zones out here. So there is a nuclei associated with each of the zones within the cortex, and within the vermal portion is the vestigial nucleus located right here, and the vestigial nucleus within the vermis is going to be involved in modulating postural adjustments of body extensor muscles that are mediated by the vestibulospinal tract, and it will help coordinate head and eye movements that are mediated by the vestibular nuclei. In the paravermal zone, we have the interposed nucleus, and this nuclei is going to be involved in coordinated movements of the proximal extremities. And then in the lateral zone, there is the dentate nucleus that is involved in coordinating voluntary control of the distal extremities. So if you draw a little stick man on here, you can see that the vermal zone would coordinate the extensor muscles of the trunk for posture as well as head and eye movements, whereas the paravermal zone would be involved in coordinating voluntary movements of the proximal muscles, like within the shoulder, whereas the lateral zones would be involved in coordinating voluntary movements of the distal extremities. And this picture down here just shows that these deep cerebellar nuclei are located within the medulla of the cerebellum, which is basically at the bottom of the white matter, so they're kind of like how the basal ganglia is within the white matter of the cerebrum. It's the same sort of thing. There's one exception, but the deep cerebellar neurons are going to be the only neurons that project out of the cerebellum, so they're going to be the only ones that provide excitatory projection output. These nuclei are going to be excited by the climbing and mossy fibers that are coming from different areas of the cerebrum and the spinal cord, and they are going to be inhibited by Purkinje cells that are coming out of the cerebellar cortex. Remember that the Purkinje cells are going to be the sole output from the cerebellar cortex, and they are always going to be inhibitory. So while the Purkinje fibers are the sole output from the cerebellar cortex and they are inhibitory, the deep cerebellar nuclei are the sole output from the cerebellum and they are excitatory. So first, let's just take a look at how the climbing fibers and the Purkinje fibers all interact to excite and inhibit the deep cerebellar nuclei. So within the cerebellar cortex, we have 3 layers. We have the molecular layer, the Purkinje layer, and the granular layer. And the molecular layer is going to be most superficial. So deep to the granular layer would be the medulla, which is where the deep cerebellar nuclei would be located. So the climbing fibers are from the contralateral inferior olivary nucleus, and only from the contralateral inferior olivary nucleus. So they're going to enter the cerebellum and provide a collateral branch to the deep cerebellar nuclei to excite it, which is going to allow the deep cerebellar nuclei to provide excitatory output outside of the cerebellum. So before we talk about the next synapse from the climbing fibers, I want to talk about these Purkinje cells, which are located in the Purkinje layer of this cerebellar cortex. So their cell bodies are in the Purkinje layer, but their dendrites stick up into the molecular layer. So the climbing fibers are going to reach all the way up to the molecular layer to excite these Purkinje cells, which are going to allow the Purkinje cells to provide inhibitory output to the deep cerebellar nucleus, which will turn the deep cerebellar nuclei off. Which is kind of confusing because the climbing fibers are going to excite the deep cerebellar nucleus and then indirectly inhibit it through the Purkinje fibers. But you have to understand that it is happening via a timing circuit to allow for coordination of the body. So I want to reiterate right now that these Purkinje fibers are going to be the only or the sole efferent output out of the cerebellar cortex, and they are going to be inhibitory. So the mossy fibers are going to be made up of input from all other places that give input to the cerebellum. So those could include pontocerebellar fibers, the spinocerebellar tracts, vestibulocerebellar fibers, reticulocerebellar fibers, and trigeminocerebellar fibers. And these are a little bit more complicated. So the mossy fibers are going to enter the cerebellum and then excite the deep cerebellar nuclei via a collateral branch, just like the climbing fiber did. And then the mossy fibers are going to go up into the granular layer and excite a granule cell. Granule cells are the smallest and the most numerous type of neuron in the central nervous system, and they are going to be the only neuron within the cerebellar cortex that provides excitatory input. All of the other neurons located in the cortex, meaning up here, with their cell bodies located there, that is, are going to be inhibitory. So obviously that's not including the climbing and the mossy fibers, whose cell bodies are not located in the cerebellar cortex. So these granule cells are going to provide indirect excitatory input to the cerebellar cortex. So they are going to project their axons into the molecular layer, where they are going to form these parallel fibers. The parallel fibers travel in the direction of the transverse axis of the folia, where they will synapse with the dendritic trees of the Purkinje cells. So just like the climbing fibers, the mossy fibers are going to come in, excite the deep cerebellar nuclei, then excite the granule cells, which will excite the Purkinje cells, which will inhibit the deep cerebellar nuclei, turning it off. Also in the granular layer, we have Golgi cells, so G and G. These Golgi cells have dendrites that extend up into the molecular layer, and the granule parallel fibers will synapse on the Golgi cells to turn them on, and then the Golgi cells will then inhibit the granule cells. This allows the Golgi cells to modulate the excitatory output of the granule cells and provides yet another timing circuit. So far you can see that within the granule layer there are two types of cell bodies, the granule and the Golgi cells. In the Purkinje layer, the only cell bodies are the Purkinje cells, and then in the molecular layer you have multiple dendrites from the Purkinje cells and the Golgi cells, as well as parallel fibers from the granule cells. Also, there are the axons from the climbing fibers. But in addition, there are two types of cell bodies that will be located there, and those are the stellate and the basket cells that provide inhibitory output to the Purkinje cells. So as you can see, most of the cell bodies that are located within the cerebellar cortex are inhibitory, and they provide inhibitory output via GABA, the transmitter, whereas the only cell that provides excitatory output, who has a cell body that lives within the cerebellar cortex, is these granule cells. So keep in mind, all of this excitation and inhibition is going on to modify the output of these deep cerebellar nuclei, which are the fastigial, the interposed, and the dentate nuclei associated with the three zones that I talked about earlier. So now we're going to talk about the pathways that are associated with each in particular zone. So first we're going to talk about the vermal zone, vermal zone, which is located within the midline of the cerebellum. So keep in mind that this is associated with the vestigial nucleus that is located within the white matter of the cerebellum. And remember that this is involved in postural adjustments of extensor muscles via the vestibulospinal tract and coordinating head and eye movements mediated by the vestibular nuclei. So the vermal zone is going to receive information about the position of your body and proprioceptive input from the vestibular nuclei, and it's going to receive information from both vestibular nuclei, so it's going to be bilateral, except one is going to provide excitatory input on the ipsilateral side, whereas the contralateral side will provide inhibitory input. And then it's also going to receive this information from the spinocerebellar tract and the contralateral inferior olivary nucleus. So the inferior olivary nucleus is going to provide a climbing fiber to the cerebellum, whereas the other three are going to provide a fiber that is a mossy fiber, because everything that's not a climbing fiber would be a mossy fiber. So the climbing fiber is going to travel into the cerebellum and synapse at the vestigial nuclei, turning it on, which is a deep cerebellar nuclei, and then it's going to travel up into the molecular layer and excite the Purkinje cell, which is going to go back to the deep cerebellar nuclei and turn it off. We're going to see the same thing from the vestibular nuclei, except this is going to happen via a mossy fiber, so this would actually be happening through a granule cell, but I'm not going to illustrate that every time. And we'll see the same thing via the spinocerebellar tract. The contralateral vestibular nuclei would provide inhibitory output, so that would be a little bit different. So keep in mind that this is receiving all of its afferent info from either the spinal cord or the vestibular nuclei or the brainstem, so none of this is coming from the cortex. Therefore, most of this is involved in more reflexive movements. Nothing that this does should be conscious, which will be different for the other two tracts that are going to be integrating information from the cerebral cortex. Then the vestigial nuclei are going to project to the ipsilateral vestibular nuclei to excite them, which will obviously give rise to the vestibulospinal tract within the spinal cord. Additionally, you should know that these excitatory fibers that are coming from the vestigial nuclei are going to leave the cerebellum via the inferior cerebellar peduncle. So overall, you can see that the vermal zone is going to integrate information from bilateral vestibular nuclei, the same side spinocerebellar tract, and the contralateral inferior olivary nucleus to get information about where the body is to make postural adjustments. And it's going to do that by exciting the vestibular nuclei to go down to the vestibulospinal tract. And it'll also, via the vestibular nuclei, will correct head and eye movements based on the position of your head. And we'll talk about that a little bit more later when we get to the vestibular lecture. So now we're moving into the paravermal zone, which is going to be involved in coordinating voluntary movements of the proximal muscles of the extremities. So when we're drawing our little stick man on the cerebellum, these would be the muscles located in the proximal extremities, obviously. So the paravermal zone and the nucleus interpositus is going to receive information from the contralateral cerebral cortex, the contralateral inferior olivary nucleus, and the ipsilateral spinocerebellar tract. So once again, the inferior olivary nucleus is going to be a climbing fiber, whereas the pontine nuclei and spinocerebellar tract are going to give rise to mossy fibers. So the climbing fiber from the contralateral inferior olivary nucleus is going to go into the cerebellum, give rise to a collateral branch to excite the nucleus interpositus, and then it's going to go up into the molecular layer and excite the Purkinje cell, which will go down to inhibit the nucleus interpositus. The contralateral cerebral cortex is going to activate the pontine nuclei and the pons of the brainstem, which is going to give rise to a mossy fiber that's going to enter the cerebellum via the middle cerebral peduncle, and then it's going to again activate the nucleus interpositus or excite it, and then it's going to go up and also excite the Purkinje cell. And once again, it's through a mossy fiber, so this would actually be through a granule cell. The spinocerebellar pathways are going to give rise to mossy fibers that are going to enter the cerebellum, activate the nucleus interpositus, and then go up to activate the Purkinje cells. So as you can see, this zone is receiving information from the cerebral cortex, so it's coordinating voluntary movements of the proximal muscles. So we have afferent info from the contralateral cerebral cortex, the contralateral inferior olivary nucleus, and the ipsilateral spinocerebellar pathways. So once the paravermal zone integrates information from the contralateral cerebral cortex, the contralateral inferior olivary nucleus, and the ipsilateral spinocerebellar pathways, the nucleus interpositive is going to project efferent info to the contralateral thalamus and red nucleus, so that you can end up affecting pathways on the same side as the nucleus interpositus. So the nucleus interpositus is going to project to the thalamus and the red nucleus, and that's going to be via the superior cerebellar peduncle. And there's actually going to be a decussation in the superior cerebellar peduncle that allows it to affect the contralateral thalamus and red nucleus. And then the thalamus is going to excite the cerebral cortex that's on the opposite side of the body, so on the contralateral side of the body. And then the cerebral cortex will obviously give rise to the corticospinal tracts that will decussate in the medullary pyramids and end up affecting muscles on the same side as the nucleus interpositus in that paravermal zone, which is how the cerebellum affects the same side of the body. And we know that the same thing will happen for the red nucleus because the rubrispinal tracts decussate in the midbrain. Lastly, we're going to talk about the lateral zones of the hemispheres, which is going to be involved in coordinating control of the distal extremities. And this should actually say lateral zone here, because the lateral hemisphere comprises both the paravermal zone and the lateral zone. So you have the lateral hemisphere, which is all of this, and then the vermis and the cerebellum. And the lateral hemisphere makes up both the paravermal zone here as well as the lateral zone here. So sorry if that was confusing. So the nuclei within the lateral zone is the dentate nucleus, and the primary afferent input to it is going to be from the contralateral cerebral cortex. So the cerebral cortex is going to project to the pontine nuclei, which are going to project into the cerebellum. These are going to be called the pontocerebellar fibers. So because it's from pontine nuclei, it's going to be a mossy fiber, which is going to go in and excite the dentate nucleus, which is going to go up into the molecular layer and excite the Purkinje cell, which will come back and inhibit the dentate nucleus. And just to reiterate, because it's a mossy fiber, this will actually be projecting via a granule cell in the cerebral cortex. So once the dentate nucleus receives info from the cerebral cortex, It's going to project once again to the contralateral thalamus and contralateral red nucleus, and it's going to do that via the superior cerebellar peduncle, just like it did for the nucleus interpositus. And it's going to decussate in the superior cerebellar peduncle. And then, once again, the thalamus excites the cerebral cortex, which will give rise to the cortical spinal tracts that will affect the side of the body that is on the same side as the dentate nucleus, but on the contralateral side of the cerebral cortex. I should point out that this pathway has been implicated in the motor learning of complex motor patterns, so this pathway would be utilized by someone who was doing something complex like playing the piano. So just to reiterate, this lateral zone that's involved in the motor planning of distal extremity movement is going to receive information from the contralateral cerebral cortex and project through the pontine nuclei to excite the dentate nucleus, which will project back to the cerebral cortex via the VA-VL thalamus and the red nucleus to end up affecting the extremities that are on the same side as the dentate nucleus. So now I'm just going to talk about the cerebellar feedback loop, which is how output from the cerebellum is relayed back to the cerebellum via two relay nuclei, which are the red nucleus and the inferior olivary nucleus. So the dentate nucleus from the lateral zone and the nucleus interpositus from the paravermal zone both project to the red nucleus via the superior cerebellar peduncle. And there's going to be a decussation in the superior cerebellar peduncle, which allows it to excite the contralateral red nucleus. And then the red nucleus is going to project to the inferior olivary nucleus, and that's going to happen via the central tegmental tract. And then the inferior olivary nucleus is going to project back to the cerebellum, and it's going to do that via the inferior cerebellar peduncle. And a lesion in the central tegmental tract will result in a degeneration of the inferior olivary nucleus, which will cause contralateral palatal myoclonus, which can be described as a oscillatory, clicking of the soft palate. And these fibers that are in the central tegmental tract that are traveling from the red nucleus to the inferior olivary nucleus are called rubro-olivary fibers. So I just want to point out that the red nucleus receives information from the contralateral dentate and nucleus interpositus via the superior cerebellar peduncle, like I've already said. But these fibers would be called cerebello-rubro fibers because they're projecting from the cerebellum to the red nucleus. But in addition, the red nucleus also receives information from the cerebral cortex, the ipsilateral cerebral cortex. So these fibers would be called cortico-rubral fibers. So we already know that one function of the red nucleus is to give rise to the cerebrospinal tract, but in addition to that, its job is to integrate information from the ipsilateral cerebral cortex and the contralateral cerebellum and integrate it and relay it to the ipsilateral inferior olivary nucleus. The inferior olivary nucleus is another site of motor and sensory integration. We've already stated it's going to receive info from the red nucleus via the rubro-olivary fibers, but it will also receive afferent information from the cerebral cortex. It will also receive afferent information from vestibular nuclei and information from the contralateral spinal cord via a spino-olivary tract. And once this inferior olivary nucleus receives all of this afferent info, it will project to the contralateral cerebellum via the inferior cerebellar peduncle, like we've already stated, and it's going to go to basically all regions in the cerebellum as a climbing fiber, because we know that climbing fibers only originate from the inferior olivary nucleus. These could also be called olivocerebellar fibers that will branch in the cerebellar cortex to form several climbing fibers, and each climbing fiber will terminate onto a single dendrite or an apical dendrite of a single Purkinje.